I'm Sergiu Pașca. I'm a professor of psychiatry and behavioral sciences here at Stanford University. My lab is interested in understanding the biological basis of brain disorders such as autism and schizophrenia. Let's walk into the lab and show you what we do.
How would you explain your work to a fifth grader?
Well, we study the human brain, and we're trying to understand what makes the human brain unique and what it makes it uniquely susceptible to disease. And so one of the challenges has been that the human brain is inaccessible. It's not an organ that you can just easily go take samples from, study the biology of that condition. Essentially what we do is we take skin cells from patients that have autism or other neurodevelopmental conditions, and then we turn them into stem cells in the lab and then literally guide them to become brain cells. That allows us in a non-invasive way to actually ask questions about the biology of these conditions, and our hope is that soon we'll be able to develop effective therapeutics for these conditions.
What's one thing you wish more people understood about your work?
I wish more people would understand why we're doing this type of work. In psychiatry and neurology, we have drugs that can sometimes help with some of these conditions, we can manage them, but we don't really have cures. One of our goals has been to try to understand the biology of this psychiatric condition so well that this therapeutic development will come naturally.
Do you have a favorite aha moment in science?
Oh, I have several. And actually the first one was on this microscope right here. Precisely 17 years ago, we got some of the first stem cells derived from patients with autism, made some of the neurons, and I remember very vividly the moment when we started imaging the neurons and we saw how they were responding to electrical stimulation. It was really, an unbelievable moment. This is a cell culture room. This is where we grow some of the cells, and we keep the cells here in this large aggregates of neurons and glial cells, which are also known as organoids or assembloids.
When do your best ideas come to you?
Actually, they come when I talk to my students.
A small daily habit that keeps you grounded.
Meditation.
What keeps you curious?
I'm curious about everything, all the time.
What's the broader impact of your work?
There have been implications of the work that we do that go far beyond stem cell biology, no doubt, but even far beyond neuroscience. For instance, we use cells from patients, and so there is this ongoing discussion of how consent should be done, for instance, for some of these patients who are donating cells. And I think there are even broader implications. They're almost philosophical at this point. And that's interesting because that allowed us over the past few years to engage more and more of the people on campus, not just in the medical school here, but really to the law school, having discussion about the ethical implications, in the engineering school about getting the systems more and more complex, and even in the Hoover Institution about the broader policy implications of the work that we do.
What's the wildest dream for the impact this work can have?
That the work that we do, the technology that we develop, will ultimately demystify psychiatric disorders.
How do you unwind?
Art. I love art. I love walking in museums. Every time I travel, I always spend a couple of hours in a museum, especially in Europe. It's kind of the way I disconnect from what I do daily.
Final question, a quote you live by.
I like what Sidney Brennan once said, that innovation is an assault on the unknown.